Welcome everybody to this first presentation of this joint POMP and SEA-Quester seminar series. My name is Michael Sejer and I'm the coordinator of POMP. Today is the first of 6 planned talks that we have on the program for this spring, one approximately every 4 weeks. For each seminar, there will be a single presentation by a speaker, and then there will be time for questions afterwards. So if you have any questions that you would like to raise at the end of Dorte's talk, use the Q&A option at the bottom of your screen. And then I would also like to remind everybody that we are recording this and hopefully posting it online for other people to enjoy also. But today it is my privilege to introduce Professor Dorte Krause-Jensen as our first speaker. Dorte is a close colleague and friend of mine through more than 25 years. And she has been involved in numerous seminal papers on blue carbon in general and on the role of Arctic kelp in particular. So, Dorte, the floor is yours, please.
Thank you very much for the opportunity to be in this webinar, and I hope you will have some good discussions afterwards. Yes, so the background photo shows a kelp forest from the Norwegian coast. And while large brown macroalgae are known for their role in stimulating diversity, then ecological functions associated with their fate in the food web and as carbon sinks, it has traditionally received much less attention, and especially so in the Arctic.
Excuse me, Dorte. Am I the only one who only sees? I don't see your presentation, Dorte.
You don't see it?
I can see it, I hope.
What about everybody else?
I can see it.
Yeah, me too.
Okay.
Uh-oh. Mikael, you can come in here and join me.
Yeah, I'll do that. Just go ahead. Sorry.
Okay. Yeah, so my interest in this field started about 10 years ago when Carlos Duarte and I, inspired by the blue carbon work on rooted macrophytes, we did a literature review to explore if macroalgae might also contribute to marine carbon sinks, although they grow on rocky substrate. So we had expected that most relevant studies would represent sedimentary sites close to the macroalgal habitat. But to our surprise, most records represented the deep sea and were very interdisciplinary, ranging from oceanography, geology, and ecography to fauna ecology. So we synthesized the findings in this conceptual diagram on the export of particulate and dissolved organic carbon from the coastal forests via surface currents and subsurface currents to shelf sediments and the deep sea. And we hypothesized that floating algae could be entrained by currents to depths where pressure caused their air bladders to collapse so that they would sink faster. And we also assumed that below 1 kilometer of depths, everything that reached that would be sequestered for sufficiently long timescales, for more than 100 years, so that it could be considered sequestered at that timescale at least. So that was how we did, and here I will further explore macroalgal blue carbon through updated studies and tests of these original hypotheses with a focus on examples from the Arctic. And my focus is on the role of macroalgae in carbon cycling rather than their potential for climate change mitigation. So, because importantly, if the pools and fluxes, they were status quo, they would have no implication for climate mitigation. A mitigation effect, or the opposite, would require changes relative to baseline. And given the challenges of quantifying status and then quantification of mitigation effects, it remains overly challenging. So there's this recent review on acceleration in macroalgal blue carbon research. And there are many different definitions of blue carbon, and well, according to the IPCC, it's the biologically driven carbon fluxes and storage in marine systems that are amenable to management. And macroalgae are in this context seen as emerging blue carbon ecosystems because they can partly be managed, but they are not yet considered a manageable system in the sense of of climate change mitigation. But overall, this shows that there's been a huge acceleration in studies on macroalgal blue carbon over the past decade, and it has been those studies have been addressing both particulate and organic fractions and also inorganic fractions, and they've been looking at fixation, burial, export, degradation, and mapping of macroalgae. So how about the Arctic macroalgae relative to this? So there's much more limited data on Arctic macroalgal blue carbon. And then we could expect that the low temperatures up there would imply slower metabolism. And also the long polar night and day would have implications for macroalgal systems there relative to elsewhere. And also regarding the future, we see this poleward expansion of macroalgae with increasing temperatures as the dominant species. They have a boreal origin with optimum temperatures way ahead of way above what they see currently. And then also the melting cryosphere will imply less sea ice, so it could mean more light to the seafloor and more habitat for the algae if this is not ruined by increased turbidity due to runoff from glaciers, which is also increasingly seen. And then another difference is that there are less people in the Arctic than elsewhere, so when talking about blue carbon strategies, then there's not so much about restoration of macroalgae in the Arctic. It would be rather looking at protection, and with the increased accessibility of the Arctic, there might be need for protection in the future. But my presentation will build knowledge on macroalgal blue carbon based on, with carbon fluxes from the habitat to the sink. And there will be examples of global scale studies that can potentially be downscaled to Arctic scale, and also of local Arctic studies that can be upscaled. And I will present examples of studies of various approaches, both in situ data compilations of habitats and blue carbon, carbon stocks, and remote sensing and GPS tracker studies of floating macroalgae, modeling studies, experiments, and biomarker studies to identify macroalgae in sediments. And then I will also show a few studies on the trends and drivers of macroalgal area and primary production, both with regard to temporal change, but also spatial gradients and predictions on that basis. So this 2016 study, we tried to come up with a first order estimate of carbon sequestration capacity at the global scale, and it was a mass balance based on estimated global production of macroalgae and the fraction of that exported beyond the habitat as particulate and dissolved organic carbon, and then the proportion of this that might be sequestered either on the shelf or in the deep sea. And these blue carbon storage estimates, they are marked in blue. And overall, we estimated that 173 teragram, or 11% of net primary production, may be sequestered every year. However, this estimate relied on very limited field evidence and called for further verification. So we gathered a group effort to jointly underpin and update the estimates. And the first task was to update the global macroalgal area and NPP, which formed the basis of all those calculations and which relied on non-supported information that had actually circulated in the literature for decades. And the updated estimates, they were in line with existing averages and indicated that global macroalgal area and NPP are in the range of that of the Amazonian forest. And then the next task was to try and improve the estimates of macroalgal particulate organic carbon to deep ocean sinks. And Karen Philby-Dexter led this this exercise. And the estimate, again, depends on the macroalgal net primary production along the shores and the fraction of that is exported from the coast. But then the model looks at the decomposition rate of this carbon that is exported relative to the time it would take for this material to be carried by ocean currents to sites where they are above the deep sea. So then it's a question of whether this material would have the time before it's degraded to reach those sites. This is what we have estimated here. So then to do that, we needed an estimate of macroalgal decomposition rates. So that was compiled from the literature, and as you can see here. And then we used an oceanographic model to compute to grab the algae and let it travel to these sites. And overall, we found that about 15% of the net primary production, or about 56 teragram of carbon, reached beyond the shelf break above 200, to where water depths were more than 200 meters, so that the algae that would sink from there, they would have the potential to reach to high depths. And we found a huge spatial variability in these estimates, with the largest macroalgal fluxes, where we had extended and productive macroalgal habitats along the shore, and where those habitats grew very close to continental shelves and marine canyons, so that the transport way to the deep sea was short. And some of even when the macroalgae reached these sites, then along with the way down through the water column, some of this would be mineralized, some of this carbon. And we combined our study with a recent study by Siegel et al., who had they had an estimate of the timescale of sequestration of CO2 injected at different depths in the water column. And using this estimate, we came up with the estimate that 4 to 44% of those 56 teragrams, or 4 to 44 teragrams in total, could be sequestered for more than 100 years at the global scale. Yeah, so this study was also, it was spatially explicit, so we could see the trends for the different regions of the globe, and also at the national scale. And for the Arctic, we could see that there was a relatively high fraction of macroalgal carbon remaining after 100 years, probably related to lower temperatures there. So this served as a first input to Arctic scale macroalgal deep sink estimates when we sum up for these Arctic countries. And our global estimate of the macroalgal NPP from this Philby-Dexter et al. Paper, it also entered this study of primary production in the Arctic by Carla Tarr, where the macroalgal production was compared to primary production of other types of primary producers in the photic zone of the Arctic. And from this, we could see that macroalgae, this estimate suggests that macroalgae constitute almost 9% of the Pan-Arctic marine primary production. So indeed, the fluxes from macroalgae are important at the Arctic scale. And the idea is then that this global model, we will downscale this to the Arctic scale, or we'll build it up again to the Arctic scale, but compiling all the newest information from the Arctic region, because a lot of data is accumulating at the moment. So we will go from the Arctic, Arctic kelp area and productivity. And there's a new area model for Arctic macroalgae being developed in the Blue Arc project, and also a compilation of Arctic macroalgal biomass. And then we will look at the decomposition rates targeted at the Arctic zone, where we expect they are lower than for the global range because of the lower temperatures. And so we'll compile this information, and Louisa Duzedom, who is postdoc in our department, she will be leading this effort. So hopefully next year I'll be able to present an updated version here with more concrete data from the Arctic. But those studies that will enter this model, they are several. And I'll just show some examples of those recent Arctic studies on macroalgal blue carbon. So there is a case study from Canada, Nova Scotia, where Kira Krumhansel and colleagues, they have estimated the fluxes of macroalgae from this area, and they have been looking at both dissolved organic carbon fluxes and also particulate organic carbon fluxes, and the fate of this carbon in whether it's exported, whether it reaches the shelf break or not, and also how much carbon is buried in seagrass meadows and in bare sediments in this area. And for this area, they actually estimate that the vast majority of macroalgal carbon sequestration is due to DOC, dissolved organic carbon export. And that overall, this constitutes about 27% of the net primary production. And we also have studies from Greenland, and there's this study led by Thomas Ayer from the Nuuk-Kunkangalua, the Nuuk Fjord system, where he measured the distribution of macroalgae in the entire fjord system, which is the greatest, one of the greatest in the Arctic, and also estimated the amount of floating algae in the same system. And the floating algae is what you can see here in green. And then in order to understand the fluxes of those floating algae and to know whether they would remain within the fjord system or whether some of them would be exported, we applied trackers, GPS trackers, and followed their trajectories. And those released in the inner parts of the fjord systems, they largely remained there, whereas those that were released in the outer parts of the fjord system, some of them made it to the open sea. And overall, about 80% of the drift remained in the fjord system. So so there was quite limited export to the open coast from this area. But actually, another study that was just published last week, it has been looking at the fluxes of macroalgae from the open coasts, including this fjord, the Nuuk Fjord system, but also along the open coasts to the deep sea, and it's looking at the potential for those algae to reach deep ocean sinks in the Labrador Sea. And so the map shows the distribution of algae with floating features along Greenland's coast here. And the approach was to then try and map floating or document the occurrence of floating algae, detritus floating from the shore. And it's Dan Carlson who led the study, and he went through 1,300 Sentinel-2 images and identified 8,000 mats of floating algae in these images. So these mats are so big that you can see them from space. So some of them were enormous. And so he did this for different seasons and to understand when were the peaks of the export. And then he also looked at drift GPS drifters again, and did a particle tracker, Lagrangian particle tracking model to understand the fluxes from the system. And the third component of this study was to simulate large eddies to look at how, once the algae had reached to the Labrador Sea, how would their their export to the depths be. And I'll just show a few examples here. So now on the map, you see examples of the tracker trajectories of those drifters. So showing that algae from the shore are connected via ocean transports to the Labrador Sea. And the model estimated that the residence time was relatively short along the shore, so that the algae could pass to the above the deep sea here, where the residence time was much longer. It was 74 days here in the Labrador Sea. And then the model on the deep convection transport showed that this transport could draw down algae to depths where gas vesicles burst and supported sinking, like we had initially hypothesized in the long ago study. But then this model also, convection transport model, also showed that there could be resuspension of a fraction of these particles that had already reached down to 2 kilometers of depth, that they could still be resuspended to the surface in some circumstances. So a fraction of what we thought would remain down there for a long, long time, it could come up to the surface. And this graph shows the percentage of particles sinking depending on their rising velocity, showing that if the particles have very limited rising velocities, small air bladders, then they were likely to sink really fast, whereas those that had a lot of updrift, they sank slower, but eventually most of this material would sink to the deep sea. Then there are also studies documenting that macroalgae actually are embedded in the sediments along the Arctic coasts and also offshore. So this study, led by Sarah Örberg, explored the eDNA of macroalgae in surface sediments around Greenland and Svalbard from many, many sites, and both close to the shore, but also at quite a distance. And in 94% of all samples, she identified macroalgal DNA, also at depths, as you can see here. So this again shows that these fluxes of macroalgae are really, really present in the Arctic and well visible all over the seafloor. Sarah also led this study that was published last year. That is a next step of the first study, where we looked at the burial of macroalgal DNA with depths. So she explored 6 sediment cores from along the Greenland west coast, and that cores that were dated and were long and old. So at specific depths, she took out samples for eDNA. And this study documented that there is a millennial fingerprint also of macroalgae in these deep cores. So 86% of all the samples had macroalgal remains, and they dated up to 2,600 years back. And then, those studies on eDNA, they only showed the presence of macroalgae, but we are now looking into whether we can document quantify the amount of macroalgal carbon that is buried in the sediments. And Louisa Dussedau, she is leading this study of digital droplet PCR analysis of selected foundation species of seaweed from Southeast Greenland and Faroe Islands. This was part of the project Blue Sea. And so a lot of sediment samples were taken in fjords and offshore in both Greenland and Faroe Islands. And Louisa analyzed them for ddPCR in top and mid and bottom sediment. Well, it's not bottom sediment, it's down to 10 centimeters of depth. And I just show here a summary result for showing that overall there was more macroalgal DNA in the samples from the Faroe Islands than from Southeast Greenland. And overall much more macroalgal DNA in the fjord samples as compared to the shelf samples, whereas we did not see much of difference between those top layers of the sediments. But yeah, so this is a very promising new technique that we will explore further. Yes. And then, yeah, in relation to this, we are also reviewing macroalgal burial rates from the Arctic. And this this table shows some rates that are already compiled, and we will add more Arctic data on this to feed into this Arctic estimate of macroalgal blue carbon. And well, our findings of more macroalgal DNA in fjord sediments in the Nuuk fjord system and also in East Greenland and Faroe Island fjords, it's in line with this global study on that shows high rates of carbon burial in fjord sediments globally. So much higher than for other habitat types. And once well, we come out from the fjords, then there's also this study from Norway showing that glacial troughs are also centers of organic carbon accumulation. So we would also expect macroalgal carbon to reach those hotspots of general organic carbon accumulation. And in the POMP project, Carmen Leiva Duenas, she's leading a compilation of polar sediment organic carbon data. And this figure shows the percentage of organic carbon in surface sediments in the samples compiled. And the next steps of this will be to document this database and then hopefully to identify drivers of hotspots of carbon storage that also is relevant for the macroalgal situation, although this is general organic carbon. Yes, and then the question about climate change mitigation of macroalgal management through protection and restoration. Yes, as I have, well, I guess it's very clear from what I showed, there are so many challenges already to document the baseline carbon sink capacity related to macroalgae. And those can be summarized as this, well, due to the spatio-temporal decoupling of macroalgal source and sink, the signal dilution in the transport path, then also these large pre-existing carbon pools out there, and would also make it difficult to, well, it's also making it difficult to understand how much of this is due to macroalgae. And also, we don't know much about the net fluxes of carbon. And in this regard, there's also this very nice new paper by Andre Visser from the sea-quester project from last year that talks a lot about these issues. And then also this large natural variability in the carbon fluxes makes it difficult to quantify. And in addition, we have this changing baseline of macroalgae with climate change, which would, so if we would want to check an effect of mitigation of, for example, protecting macroalgae in the Arctic, then we would need, if we wanted to quantify it in terms of carbon units, we would need to both quantify all the baseline, and also know about the trends in macroalgal habitats, the baseline, and then we would need to quantify the additionality and permanency of the additional carbon sink resulting from the management. So in my view, this is not currently feasible. then you could say that, yes, but there are emerging Arctic ecosystems that would be new carbon pools. So if macroalgae reach sites where they were not before, then all that you see up there would then be new and could be quantified. And yes, then that would be a quantifiable new carbon pool, but this may be compensated by contraction of habitats and pools further south. So at the global level, I don't think we can really quantify how much Arctic carbon would contribute to climate. Management of Arctic carbon, Arctic macroalgae could contribute to global carbon sinks. But yes, as I said, if we want to look at the potential for management, then we also need to know about the kelp trends, because we need to know them if we want to be able to document that we made a change in the trends. So a global study on macroalgal trends or kelp trends showed a huge regional variability that by far exceeded the global average decline, which was about 2% per year over the past 50 years. So we cannot say that at the global scale there's a clear trend of declining macroalgal habitats that need to be restored. It's very local. And also at the southern distribution limit of the kelps, we do see major declines. And then we tried to understand what are the trends of macrovegetation in the Arctic. So we did a survey, a large group of us did a survey on trends, and there were not so many. We found about 37 in general, and those showing increases are shown in green, and those with decreases in red, and those with no change in black. And overall, there was a tendency for increase over the past, since the 21st century. Yeah, so since 2000. Yes, and the same study also did a modeling of the habitat suitability for macroalgae from over a 50-year period up till about 2015, and documented that due to changes in temperature and sea ice, we would expect polar migration rates of about 20 kilometers per decade for macroalgae. And a further study of area gain, modeled area gain of potential macroalgal habitat into the future suggests that such increases may continue. But it's important to say that these models assumed that kelps would grow down to 30 meters of depth in both scenarios and doesn't consider changes in light attenuation. And several new studies have documented a major change in the turbidity in the coastal zone. So although sea ice will open up new habitats, then turbidity along the coasts due to increased runoff from land might limit the capacity of macroalgae to grow deeper. So there's a trade-off there, and we are not sure what will be the net result of macroalgal area due to future climate change in the Arctic. So in conclusion, regarding the carbon cycling and budgets of Arctic macroalgae, they form extended and very biodiverse and productive habitats with significant carbon fluxes and contributions to the biological carbon pump. So they should be embedded in marine carbon budgets, and we should continue studies to improve quantification of this. Regarding management strategies, the increased accessibility to Arctic coasts may pose a risk of disturbance of macroalgal habitats and their associated carbon sinks. So there is a case for protecting these, although we cannot quantify how much this would mean in terms of carbon benefit, but it would have multiple ecosystem function benefits. So thank you. This is it.
Thank you very much, Dorte. And yeah, I'll just remind everybody, if you have any questions, please use the Q&A button at the bottom of your screen, right next to the green share button, and then write your question in there. And I can see that Jörg Dutz has done that. So his question is, can you provide a rough estimate of how changes in macroalgae biodiversity might affect the carbon burial in the future, Dorte?
How changes in the macroalgal area or composition?
Macroalgae biodiversity.
Biodiversity. So I assume you then mean the change in the actual species or composition of macroalgae.
Yes, I think that's the...
Yeah. Many of the foundation macroalgal species in the Arctic have a boreal origin. So the intertidal algae, Ascophyllum nodosum, and the Fucus vesiculosus and Disticus, as well as several of the key kelp species, Saccharina latissima and Alaria, and several other species, they have boreal origin. So with higher temperatures, they should be able to migrate further north, so they can expand their distribution range at least. And so that I think they would contribute to more carbon sequestration further north of macroalgae than was there before, because those species that were, well, there was not so much up there already. And I don't know, then the endemic Arctic species like Laminaria solidungula, it would lose importance and also lose contribution, or there will be lower contribution of that kind of species to carbon pools. Yeah, I don't know if that answers the question.
Thank you, Dorte. And then we have Paul Renault, who asked, or he states, considerable amounts of kelp detritus is eaten by both benthic and pelagic organisms. Do the modeled degradation rates you presented include consumption or just degradation by a microorganism?
We are looking at the degradation rates in that model by Philby Dexter here. Oops, there. So we are looking at decomposition rates of macroalgae in this model. And then the likelihood that some of this will reach out there. But then, of course, some of it will be eaten.
So there's an additional loss by consumption.
So we had, in this estimate, we had production rates of macroalgae along the shores based on a global model, and then we estimated how much is exported, and we estimated that about 60% of the production was exported. So there could be some grazing that was causing this estimate to not be 100%, but then there could, of course, be grazing also along the way. And when we reduce the estimate of, well, the 56 teragram of carbon that would reach areas where it's reaching areas where it's in the sea, where it's more than 200 meters deep, then when we look at how much is might be sequestered for 100 years, and it's only a fraction of this, then it takes into account that a lot is degraded on the way. But again, we have not been able to find estimates of how much of the remineralization would be also facilitated by grazers.
Just a question to Paige, who's organizing this. Is it possible for people just to unmute and ask the questions directly?
I don't think so, but I can check.
Maybe we can just have somebody in the audience give it a try and see if they can
Yeah, I mean, I can allow someone to talk, so I just don't know if they can. It looks like they could maybe raise their hand, so we could try that if they wanted.
So just to say, if there's additional questions and you would like to ask them directly to Dorte, we think it's an option. Just use the raise the hand.
We can try, yeah.
Yeah.
And I can also stop sharing so that we can see each other. Shouldn't I do that?
I had a quick question about this slide, if you have a minute.
Okay. Yeah.
Why is the decomposition rate of the Echolonia genus so much higher than all the other ones?
Echolonia. That's a good question. I don't know, actually. Maybe it's because it's in regions that are warmer. So yeah, if it's in situ estimates at in situ temperature, then that might be one reason. But we have not been looking into reasons for differences between them. But for the Arctic updated Arctic study we will extract those species that are relevant for the Arctic zone. So so then we would tend to be in the range here of lower decomposition rates.
Thanks.
Okay, thank you, Dorte.
Oh, it looks like Chris raised his hand, so I'll see if I can.
I will stop sharing here so that.
Yeah, hopefully you can talk now, Chris.
Good to see you.
Hello, yes, thanks for an excellent talk. I was wondering, since macroalgae contain carbon, nitrogen, and phosphorus, what are the implications of these immense pools in terms of nitrogen? That must have implications for how the benthic food web works, and since it receives potentially in some hotspots quite a lot of nutrients as well.
Yes, I think that's really important. Also because during the summer period, and especially the late summer period, there's nutrient limitation in many places up there. So we studied actually the fluxes of ascophyllum in connection with the annual reproduction. And we hadn't thought that reproduction would mean a whole lot in terms of carbon fluxes of ascophyllum. We've been looking at growth, vegetative growth, and it was only because we then arrived to the Arctic at a slightly earlier one year, and we saw all those receptacles just covering the intertidal zone. It was just light brown of receptacles. So we thought, we're missing a huge part of the carbon and nitrogen fluxes by not embedding this. So then we did a study on the reproductive effort, and because it's emitted over quite short periods, then I think that flux would also add a lot to the coastal ecosystem. And it's in the late summer when, or in midsummer when there could be limitations. So I think the macroalgae do contribute a lot also to the food web, both in terms of these fluxes of nitrogen, but also as a direct food source and detrital source of material for the food web.
Thank you.
Great, thank you, Dorte. Any other questions?
I don't see anybody else's hand raised.
Okay. Well, thank you everybody for attending. Thank you for Paige for setting this up and hosting us. And thank you, Dorte, for volunteering for this first talk. I will just remind everybody that our next talk will be in about 4 weeks. There will be a reminder to everybody, so stay tuned. And thank you again for everybody for attending.
Thank you for arranging. It's a good idea to have these seminars. Yeah.
Great.
Happy New Year, everyone.
Yeah.
Happy New
you
Year.
too.
Bye.
Bye.
Bye.